So thanks for that introduction. As you said, my name is Brittney Mills, and I am from Abbvie. The title of my talk today is the use of high-throughput methods for early-stage formulation evaluation and how we can use those methods to correlate to our extended accelerated stability studies. Okay, let's see. There we go. I'm going to start just with a little bit of an outline. So I'm going to start with an Abbvie introduction to Abbvie. I'll then go into the details surrounding drug-like properties and why we are concerned with monitoring them. Then give a little bit of background on the high-throughput excipient screening that we do in my group, and then how that data that we generate compares to those accelerated stability studies. So first off, Abbvie, who we are. So we are a biopharmaceutical company that was founded in 2013. Most of you have probably heard of Abbott. So we are that break-off portion of Abbott that is more centered on those biopharma products. We currently have around 47,000 employees in over 70 countries. Our headquarters is located in Lake County, Illinois, and that's where I am based out of. But we also have sites in California, Massachusetts, as well as a site over in Ludwigshafen, Germany. Our focus areas I have listed here: immunology. I mean, many of you probably have heard of Humira. We also recently have Skyrizi that was approved as well, so very active in the field of immunology. And then we're also focused in oncology, neuroscience, eye care, virology, and aesthetics, with eye care and aesthetics lines being recent additions due to the Allergan acquisition. We also have other areas of specialty, including women's health, cystic fibrosis, and diabetic neuropathy. Okay. And then finally, we received more than 40 top great employer places to work rankings globally. So that's one kind of thing I always like to point out is, here at Abbvie, we do enjoy what we do, and we are very, very valued by the company. So with that introduction in mind, I'm going to dive into the talk and, why we're here today. So first off, the drug development process and what does that look like, and where am I situated within this this sector. So, early on in the discovery side of things, here I'm going to turn my pointer on. Okay. So, in the discovery side is kind of where we start. Those molecules are identified, looking for affinity, binding, epitopes, different things such as that. And then it goes on to preclinical development as well as the clinical development. And so I am part of the preformulation group here at Abbvie. And so we come in once we start with the drug-like property assessment. So, that is looking at different things such as conformational and colloidal stability and different parameters such as that I will go into on future slides. So we are kind of situated right in that phase, that interface between discovery and that preclinical development and that clinical development. So we aid in providing information to teams that allow for them to select both based on those, those key discovery attributes, as well as the longer term, drug-like property attributes. Is this something that's going to be stable in a vial or a syringe? And trying to help them screen molecules that would be suitable from both perspectives. So when I refer to, drug-like properties, what am I talking about? And so there are multiple different factors that we consider when we are determining if a molecule is a suitable candidate for development. The first thing we really look at is that primary sequence of the antibody. That framework really dictates the molecular robustness surrounding the molecule. And so that first primary sequence influences what we see for colloidal stability, conformational stability, chemical stability. All of these drug-like properties are really tied to that intrinsic factor of the protein. So if we identify key liabilities that we can screen out in that primary sequence, that's kind of the first step. And after that point is when we assess the drug-like properties of the molecule. So as you can see here in this schematic, there are multiple things that we consider. So within the pre-formulation group, I'm primarily concerned with looking at the conformational stability, colloidal stability, and the chemical stability. Those are the three pieces of the puzzle that fall under, my shop and the work that we do. In addition to that, the discovery colleagues look into that biological activity and see, are we getting correct in vivo binding? What does the binding epitope look like? How is that changed? And then as well as, our colleagues in preclinical safety who do, the tox studies looking at the safety of the molecule. All of those things kind of come together, those different pieces of the puzzle, come together to identify or to make up the attributes of the candidate that we're pushing forward. Shown in this bottom portion here are those things that we must consider that could influence the observed drug-like properties. So, processing conditions. For example, some molecules are really sensitive to different types of shear stress or freeze-thaw, agitation or transport. All of these things that happen during the life cycle of a candidate as we go, from making that candidate to dosing it in the clinic, we have to consider all of those in our design. Also solution conditions, such as protein concentration, with the different new modalities that are being investigated with biologics, you have just the standard MAB, which, usually is okay to be concentrated to a high concentration, but at other times, you have other modalities such as antibody drug conjugates or different types of bispecifics or fusion proteins, and all of those have different limits when it comes to protein concentration that, are really dependent upon the DLP of the molecule. And pH, ionic strength, those are all, again, influenced by that intrinsic primary sequence, affects the isoelectric point or that neutral point, and influences the pH that we formulate at. And then administration, when you think about in the clinic, when you are, for example, for a conjugate and you dose it or dilute it into an IV bag, are you having any absorption at the low concentrations you're dosing? Are you having any aggregation upon dilution into saline? Those are all things that we must consider early on to allow us to move the best candidate to move forward. So we perform this assessment because we want, the best molecule moving forward and not just an average or an okay molecule as we go through our pipeline. So here is just a schematic of our current biologics screening funnel that we use here at Abbvie. So, really early on, before we even get involved, that's when the discovery colleagues are trying to find the appropriate binding epitope, binding affinity, doing all the different types of in silico screening, potency, and binding assays. Once we get down to kind of the tens of molecules is when we start to get involved from the pre-formulation perspective. And at this point is kind of when we implement the use of high throughput tools such as the Prometheus that I'll go into a little bit later, where we are able to just screen molecules very rapidly and get key critical data outputs that can help us to minimize or decrease the number of candidates moving forward. Once we get to around less than 5 candidates is when we do more of our comprehensive physical chemical screening. So, we pick a low and a high concentration, really influenced by the nature of the biologic modality. Look at the colloidal stability, for example, for oncology, or for antibody drug conjugates, looking at the drug to antibody ratio and different factors such as that. And at this stage too is when we'll start to kind of look into initial excipient assessments. So how does our platform formulation work? Are we going to have to add different types of excipients to stabilize the molecule, etc.? And then once we get down to that 1 or maybe even 2 candidates, that's when that formulation development really kicks in, and that's when we do excipient screening. And this is another kind of area where, the Prometheus falls into play is, excipient screening, you can test more samples, more excipients with a lower material requirements required of the instrumentation. And so we will use the Prometheus for a lot of the different types of excipients that we screen as we're trying to identify the suitable formulation. So here on this next slide shows, the analytical tools that we use to screen biologics and assess those drug-like properties. So, I don't think it's anything that is that novel compared to what is used across the industry. We look at monomer and colloidal stability. I'll show some SEC data today in this presentation. Also look at particle size, be it MFI, horizon, or different types of techniques such as that. Charge variance using standard techniques such as ICIF. For conjugates, we look at that drug to antibody ratio. And then here, I've shown here in the conformational stability, I'll go into some DSC data as that is kind of what we had commonly used before the Prometheus and how the DSC data compares to that more high throughput method with the Prometheus. So why do we characterize the drug-like properties of the molecule? Well, this assessment really serves to answer those early developability questions, around those traditional antibody formats and those novel formats, such as the fusion proteins or the bispecifics, those ones that aren't as plug and play, that require more extensive development or insight into their characteristics. So this diagram just kind of shows how everything comes together. So first off, we really look at that primary sequence to identify if there are any key liabilities that are present just due to the nature of the sequence, and if it is a possibility to engineer those hot spots or sites out of the sequence. After that point, we do a quick screen on plasma stability to see how does that molecule look with regards to aggregation or drug to antibody ratio in the instance of conjugates when it is incubated in different types of plasma. After that is when we go into, that conformational stability assessment as well as that colloidal stability assessment. So looking at the secondary and the tertiary structure, throwing them under some different types of conditions and evaluating the colloidal stability, these will really give us insights into the manufacturability as well as the shelf life potential for the molecule. For some of the formats such as, standard antibodies, typically it's been, kind of platform 100 mg per mil. Now we're being challenged to go even higher in concentrations. So we do need to look into the solubility, whether that be, low viscosity at high concentration, what the sequence looks like to see if that we have the adequate, charge repulsion to enable those higher concentration formulations. So solubility is becoming something that is getting even more increased attention due to the shifting nature of the needs from the clinic. And so this whole kind of assessment really gives us insight into our compatibility with our platform methods. How does the molecule look in our platform formulation? And then that will allow us for kind of an assessment of the speed to the market. Is it something that's going to require extensive development activities? Then we'll have to add months into the development timeline. Or is it a molecule that can kind of follow our platform processes and we can just kind of push forward? So with these in mind, I'm going to go into how we implement high throughput screening within our evaluation and our workflow. So, here I have shown, which I think we saw earlier, the Prometheus is the kind of the workhorse for a lot of our high throughput screening techniques. So really the goal of the study I'm going to describe today is to provide guidance for our formal formulation declaration. One of the big requirements for us was that we needed a technique that used low sample volume and material requirements. And so with the Prometheus and the capillaries, I think we can get away with between 12 to 15 microliters, and the concentration range varies quite widely. So it makes it very diverse there from that perspective. Also, it has a fast analysis time. It takes about an hour and a half for 24 samples, and you can get multiple data outputs, both the conformational stability as well as that aggregation propensity upon unfolding. But the question we always ran into from later stage groups was, is this really predictive for our long-term stability studies? Is the data that we're generating really helpful when it comes to making that selection? Would we miss a candidate and stop a candidate based upon this data that would have been okay from our accelerated stability studies? So that's one question that we needed to answer. So this just shows the data outputs with the Prometheus. And so on the left panel here, just shows that intrinsic fluorescence, that F350 over 330, so that unfolding event that occurs. And then here in the right panel is that T-ag. So at which temperature does unfolding lead to aggregation? And so for this one, you can see that sometimes aggregation occurs by that increase in scattering, whereas in some cases it doesn't happen. But during our traditional, formulation assessment, we get all of these data, all this data. And the big thing is with all of this data that's generated, how does it compare to standard techniques that have been commonly used? And how do we go about interpreting this data? So to start off is a comparison between the DSF, which we use with the Prometheus, versus DSC, which was our standard technique that we used for conformational stability evaluation. And I just put together kind of a little comparison here, for the Prometheus. As I said before, it requires a very little amount of sample. For our standard TM determination, we typically run them at 1 mg per mil. DSC, that requires about 500 microliters, and so that's about half a mg of material required for each condition, so quite a bit. Time is also a large difference between the two. If I were to run 24 samples on the DSC, it would take at least 2 days, if not more, based upon buffer blanks. And also the waste generated, I think it's not something we always consider, but with the Prometheus, the waste is very minimal. It's whatever is stored in your in the capillaries, whereas with the DSC, you have extensive washing of the cells and different things like that, and we really need to monitor our waste and change out the waste containers if we are running, a full set of series of samples using the DSC. So how does the data compare? Shown here on these slides, you'll see in the blue is DSF, in the teal is DSC. And so from this data, you can see that usually the TMs overlap fairly well. Because there's a difference in the technique, you may see differences in the data intensities. For example, with the Prometheus, you're looking at a fluorescence event, whereas with DSC, you're looking at a calorimetric event. So, an energy-based event. So something that requires more energy to unfold may have a different amount of fluorophores within that region. So those are, some of the reasons why, for example, in this one, we see higher intensity in the fluorescence. There was more fluorophores in this region, and it did not directly correlate to the energy associated with unfolding of that event. So, there may be slight differences. We observe just intensities, but overall the TMs seem to be very comparable, and the trends are very comparable. Oh, wrong way. So here's just a comparison, and this is just for the data set I have that I'm showing today. We've generated a lot more data comparing the two techniques, but overall the TM1s seem to overlap very well between the two techniques. There is a little bit of difference between the TM2s. For example, right now, I'll go back to this slide, for some of these, with our DSC data that we currently use, we're able to deconvolute into two separate peaks under this Fab CH3 peak, whereas there's sometimes with DSF, we just kind of take the peak intensity at the top, or the TM at the top, and we don't do the deconvolution. So that might be some reason there's a little bit of differentiation here, is because we currently do not deconvolute for the DSF data based upon the software that we have. So what is the ideal concentration to use on screening studies? This is a question I get quite often. At the stage where we implement these studies, we're usually material limited. So lower concentration would allow for more conditions to be tested. But a useful concentration really varies by the type of modality. For example, antibodies greater than 100 mg per mil, where with novel formats such as ADCs or fusion molecules, you could be much lower. But really, the screening conditions should be predictive of where you're what concentration you're going to store your molecule at long term. So we started with just kind of looking at how is the TM influenced by concentration, and overall there's not really any difference. We saw again more scattering at the TM2, just again probably due to the variation in the peak picking event, but the TM1 is very consistent between both methods. Next, we looked at the T-ag by backscattering. So essentially, as that molecule unfolds, you expose those hydrophobic interior residues, and then aggregation occurs. What we saw in these studies, it makes sense, theoretically when you think through it, that you have a higher T-ag at lower concentration than higher concentration. So T-ag is concentration dependent. You just think about it in solution, if you're at a higher concentration, your molecules, are more likely to interact and be closer together, therefore facilitating that aggregation event. So commonly, we'll see when we run these T-ag measurements at low concentration, we will not see any sort of aggregation event. But once we spike, increase that aggregation to, or increase that concentration to a higher value, we do see aggregation. So, for example, in this panel, of these formulations we tested, if we used 1 mg per mil, it all comes back green. Everything looks great. But if we test them at a more representative concentration for what we would use long term, we can see that there definitely starts to emerge, a rank ordering starts to emerge between the samples just because of the concentration. Finally, I'm going to go into high throughput screening compared to our accelerated stability studies. So I'm just going to kind of go through this slide quickly, but essentially, we choose different classes of excipients based upon the nature of the aggregation event that we are trying to limit. So, in this study here that I'll show, we have a couple structural stabilizers that work by preferential hydration. We also have some salts that I'm going to show, and some other different classes of molecules. So just for time, I'm just going to kind of bypass this slide. So it's not super important for the discussion. So for this study design, we used an antibody drug conjugate that had a DAR of 2 with a PI of around 8. We chose 7 different excipients at 2 pH values for a total of 16 formulations. We assessed conformational stability by DSC and DSF. That's just how we generated the data set that I showed earlier. And then we also did more of a traditional stability assessment where we formulated it at 25 mg per mil, stored it at accelerated and baseline conditions, and then analyzed over an 8-week period. So the first thing I'm going to show here with some of the Prometheus data is the influence of pH on the conformational stability. So what we can see is that in general, at a lower pH, we do see a decrease in that TM1. And that is a pretty commonly known phenomena, is that, that lower overall charge does cause a higher conformational stability. Usually when we decrease pH, we will see a reduction in that TM. So that was nothing that was super surprising from our perspective. There is one excipient 5 for whatever reason is an outlier, and we do see that kind of in all the data. The trends are reversed for excipient 5 in comparison to the rest. So this panel just is comparing the different classes of excipients. So we used excipients, what we classify as complexing agents. So in general, those complexing agents, as well as the structural stabilizers, shifted that TM to the right. And we can see that here in this Prometheus DSF data that, without the excipient, and then with the excipient, we do see a shift. In the excipient 3, 4, and 5, which are the salts, and that's what's shown here in this panel down here, that we add that salt and it does cause a left shift. So salts in this instance were destabilizing to the CH2 domain within the protein. Next, I'm showing the unfolding induced aggregation, so that T-ag event. One thing that I like to do when I'm trying to compare the data and look at the data is overlay that first derivative, that fluorescence data with the T-ag data, and I think it kind of can help show a little bit more detail into what's going on during that unfolding. So when you line those up, you can see that in some cases, that unfolding induced aggregation occurs, right as soon as that Fab domain is unfolding, whereas in this case shown here on the right, it does not occur until, that Fab is almost completely unfolded. So, I just kind of did a plot here that shows, which domains are more responsible for that unfolding induced aggregation in comparison to others. So one way you can kind of play with the data and understand your molecule a little bit better. Here is more T-ag data, going back to that excipient screening study that I was discussing. So with those complexing agents, we can see that at the low pH, which is that 5.5, when your molecule is more charged, we have very little aggregation events, both of the complexing agents and structural stabilizers. Aggregation only happens when we bump that pH up to 7 and we're closer to that PI. Whereas in the case of the salts, it's much more varied. Again, that theoretically makes sense. Those salts are providing kind of a shielding effect to those charged residues, which then would allow for more interaction and hence more aggregation. So finishing up here, so this is our accelerated stability data. So on the panel here up at top is all of the different different time points. So across the board, 4C, very little aggregation versus T0. But we can see after 8 weeks, there is an increase. 3, 4, and 5, and 11, 12, and 13 are all the salts, and that's kind of broken down here, where you can see that salts definitely caused a lot more aggregation in comparison to those complexing agents and the structural stabilizers. Here shown on the right is buffer alone. So those structural stabilizers and even these complexing agents were even better in this instance. I'm going to just go through pass by the DAR data because no, we didn't really see any significant trends, and kind of get to the how does it all connect to finish up here. So that's kind of our big question, right, is are our early screening studies really indicative of what we see later stage? So down here in this bottom panel is kind of more of our accelerated stability study, whereas up on top is our early screening. So T-ag at 1 mg per mil, green across the board. So we really have realized that this is not, the low concentration T-ag is not really indicative of anything for later stage. But if we do look at the T-ag at a concentration that's more relevant for our accelerated stability studies, we see that it does overlap quite well with our aggregation by SEC. So we can see that it would have, given us caution for a couple different ones that are actually okay, and that's fine. I'd rather it be that way than for it to say a molecule's good and then it actually was poor. So it does give us a little bit of an indicator early on about how those properties correlate. And so as well, the DSC and the DSF data, again, very, very similar. The red ones up for DSC were also read by aggregation, so gives us a little bit of a link between the two. And this, again, just to this specific study, what we saw is pH 7 has higher aggregation. Makes sense. We're closer to that PI, more likelihood for interaction without that charge, whereas pH 5.5 gives us worse conformational stability. Again, something that we've commonly seen in the literature. So it's really up to selecting that optimal formulation that really achieves that balance between both that conformational and colloidal stability. So in summary, the conformational stability data that we generate by DSC and DSF provides similar results. It does allow for us to rank order our high throughput, get a similar rank order from our high throughput screening methods as we do from our more accelerated stability methods. But we do need to use a similar concentration in that high throughput screen as we would for our later stage accelerated stability studies. For this ADC in particular, we did see that the complexing agents and the structural stabilizers provided the largest benefit, and that the pH of 5.5 was beneficial from an aggregation perspective, but not fully from a conformational perspective. So we really need to, select that suitable formulation that really allows for us to get the pros and cons from both sides as we achieve that formulation. So just a few acknowledgements at the end here from within my group. Melissa Fernandez helped to collect some of those data, and then for MBE Analytical R&D helped with the mass spectrometry analysis. So with that, I'd be open to any questions.